Okay, now it's time for our next lecture. The topic of our next talk is materials for biomedical applications, which will be presented by our respected speaker, Dr. Suchetan Pal from IIT Bhilai. Welcome, sir, on behalf of organizing committee. Sir has joined with us.
Okay, am I audible?
Yes, sir, you're audible, sir.
Yeah. Good morning.
Good morning, sir. Okay.
So you can see my screen, right?
Yes, sir. It is visible, sir.
Okay, should I
Okay.
start?
Please, I want to give a small
Okay,
introduction to our participants.
okay.
Okay. So our next speaker is Dr. Suchetan Pal, sir. He is an assistant professor at the IIT Bhilai. Dr. Pal has received his MSc from Indian Institute of Technology, Kanpur, and he has done his PhD in chemistry from the Arizona State University, USA. Sir has carried out postdoctoral studies in the Columbia University and Memorial Sloan Kettering Cancer Center, USA. His research expertise lies in the area of nanoparticles, bio-nanotechnology, and the self-assembly. He has co-authored more than 20 research publications in the various international journals. Really, we are honored to have you with us, sir, and we are waiting to hear from you.
Okay.
So
Thank you very much.
now I request Mr. Dr. Pal, sir, to start his presentation.
Okay, thank you very much for the kind introduction and good morning to everybody.
Good
Hope
morning, sir.
all of you are safe. And I wish this conference should have held in the campus itself. I love to visit your campus, but nonetheless, this is a nice platform so that we all can discuss about the materials in biomedical applications, right? So, as I understand, this conference is about materials modeling and mostly physical aspects of materials. For example, theoretical aspects, as well as the previous speaker said about some experimental aspects of material. So I'm trying to give you a brief introduction and outlook of materials for biomedical applications and what we are trying to do in our lab at IIT Bhilai. Right. So before I dive into my presentation, I'm very happy to share that this IIT Bhilai main campus is um, currently being built at Kutela Phata, which is very near to your campus itself, right? So in 2-3 years, we'll be moving to our new campus, and you can see that this news came very recently that IIT Bhilai is spending 700 crore rupees on this campus. So I was just correlating our materials world with this campus building thing, and I thought like most of the money, the money will be spent on materials that will be used in building this campus, right? So study of material is very important. Therefore, right? So all the theoretical aspects as well as experimental aspects of material is very important, right? So that's what this conference is about. We want to model the material properly as well as so that we have proper experimentation on the material, right? So when you go to an engineer, what happens is he will if you break something, suppose you break your computer, your phone, you bring to an engineer, he will say, okay, I know everything in the device, I know the physics, I know the electronics inside the device, and I can fix the problem, right? If there is any problem, I can fix it. Or if you give me the parts, I can actually build the device out of individual components. So he needs some tools and materials to build a device or fix a device. Right? So this is what engineers do. But when you talk about our body, you can think about our body as a very complex machine, right? So our body, I can make an argument that This is very, very complex machine, and all the physics and everything, chemistry, physics are fully not understood. So people are doing lot of research to understand the physics and chemistry in our body, right? So if you roughly think our body as a machine, right? So if you have some problem in the body, doctors can become engineer and they can fix the problem inside the body, right? Uh, Although the doctors cannot build human being, but they can fix. So if you have any problem in your body, this doctor will think that he can detect a problem inside your body, and he thinks that I have a tool to fix the problem. So he also needs some tools and materials to fix the problem in your body. Okay, so what kind of tools or materials he needs? He needs biomaterials. Not all the materials will work on your body. Only few materials out of the huge gamut of the material will work on your body. So these are called biomaterials, right? Some of the example I'm showing here in this picture is some of the examples are of biomaterials. For example, you have some problem in your eyes and you use contact lens. So this material is called biomaterials. Some people have problem in heart. Okay, so the valve, so all of a sudden stops working, the natural heart valve. So they go to hospital and they do surgery and replace the natural heart valves with some artificial heart valves. These materials made out of biomaterials, right? Some in Western countries, you can see they implant insulin pumps so that this device detects the blood glucose level in your body and they can make adjustment in insulin concentration in your blood. So they can inject insulin in your blood. So this is an example of biomaterial, right? And you have some synthetic molecules, synthetic polymers, and using that polymers, you can make synthetic arteries, synthetic blood vessels. So if you have some problem in your blood vessel, natural blood vessel, you can actually do the surgery and replace the natural blood vessel with the artificial blood vessels, right? So these are the examples of biomaterials in real life. Okay, so these materials are basically made of you can make these materials using artificial materials. For example, you can use glass or ceramic materials, um, metallic material, for example, steel, titanium, those kind of material, metal alloys. Polymers are largely used for biomaterials, and composite means you can combine two of them together or three of them together and you can make a composite material. Okay, these are the classes of artificial material. That means this origin of this kind of material is not in biology, so your body doesn't produce this kind of material. Okay, and also you can use natural materials which is produced by your body, right? Or plants, right? So you have carbohydrates, proteins, and DNA. You can use this kind of molecules to make this kind of biomaterials that can be used for any application. So it can replace some of your organs, or it can be used for wound healing application. It can be used for um, making heart valves and those kind of things people are doing. So this is the focus of huge current research, and our lab is interested in biomaterials and application of those materials in disease detection as well as drug delivery, right? So previous slides I already talked about these biomaterials. Mostly we the materials we saw is in micron scale, more than micron scale. So you can see those material in by your eyes. So materials above zero point 0.2 nanometer will 0.2 millimeter will be seen by your eye. But if you, we know all know that if you bring this material dimension below nanometer, 100 nanometer, this material property gets changed and we get emerging properties. So this is the basics of nanoscience. So we all know that in this platform, right? So this is one of the examples. So if you take a bulk gold, looks like metallic shiny gold with this typical gold color. But if you bring this dimension to less than 100 nanometer, you can see the color is changing from a golden color to purple color. And if you bring them down to 20 nanometer, it becomes this nice wine red color, right? So the optical properties of this particle material is changed when you bring it to a nanoscale, right? This is another example where this is a quantum dot, which is the light you are seeing is the fluorescence light from the quantum dot. And depending on the diameter of the quantum dot, you can actually change the emission wavelength. So you can generate quantum dots emitting blue light to the red light, and you can cover the entire visible spectrum. So my point is, you can, if you use the nanomaterials, you can have interesting property compared to the bulk materials, right? So over the years, people have generated lot of nanomaterials, and whatever bulk material you have, you can almost generate the corresponding nanomaterial with different size, shapes, as well as surface properties. Here you see there is lot of pictures. All of them falls below 20 nanometers. So diameter of each of this object are below 20 nanometer. And then see, you can develop chemistry to have palladium nanoparticle, platinum nanoparticle, silver nanoparticle, gold nanoparticle, or polymer nanoparticle, DNA nanoparticle, something like that. So in our lab, actually, we are interested in these nanoparticles made out of DNA as well as proteins, right? So we need to have something which is coming from nature. The simple reason being, these natural molecules are non-toxic, usually non-toxic to your body. So you can use these molecules for different purposes without worrying about toxicity in your body, right? So this is the primary reason we are interested in these natural um, biomaterials, right? So if you think that I have developed a nanomaterial and I can use it for biological application in real life, in clinics as well as in hospitals, that is not so easy actually. So we can do a lot of research, but getting a product out of that research is very, very challenging. So this pathway shows you how we are doing this kind of translation of these biomaterials. So we have to first design the biomaterials properly. So we have to make the material so that material composition doesn't affect your body. The chemical modification has to be done so that your material don't react to your molecules in your body. For example, doesn't react with your protein, your DNA, and so on. If it reacts with your body material, then this material is going to be toxic to your body. So that is what you don't want. Okay, and then you have to have something biophysical substitution, soluble factors. So you should not, if it is used for short-term application, then you have to have some soluble factor so that this material degrades very rapidly in your body after doing the job, right? And also cell composition, so how the cells are going on the material, um, that you have to do. All these design parameter is very, very diverse, and depending on the use of the material in your body, you modify these different things. And then after you are satisfied in your design, you go for in vitro testing. In vitro testing means you are testing the material in your lab using cells, right? So you are having cell culture and you are putting the material and seeing the activity in the cell, whether this material is toxic to the cells or is cells are doing something to the material. Is it corroding the material? Is it degrading the material? So on, different things you can study in vitro testing. Okay, it takes typically 1 year, and then after you are satisfied your in vitro testing, your material is non-toxic to the cells, you can go for in vivo studies. That means you are studying this material on small animals. right? So animal trials would be depending on the material you want to put in. So you'll do the experiment in small animals like in mice, in rats, in guinea pigs. After you are satisfied with this study, there is no adverse effect in vivo studies. Typically, it takes few years. You go for clinical testing. So you talk to a doctor, and they want to try clinical trial, and you test these biomaterials in real human patients, right? Then if it is successful, you are fine. You can go for in production of the material. But if it is not successful in any of the stages, you have to go back to the original design. You can change the design, and this is a trial and error thing. But you have some initial information which in materials to use and which is not to use, right? And right now, a lot of computation has come in. There is some um, artificial intelligence, big data analysis, those kind of things are there. Machine learning is there, so that can also predict some um, prediction like clinical outcome from this designing parameter, and they can predict what kind of design you need, right? So this is a huge area of interest, and a lot of people are working in this area, and our lab in IIT Bhilai is working in this direction, right? So in IIT Bhilai, I am working with three excellent independent researchers, Akbar, Shaista, and Saipawan. They are working in my lab. And basically what we are doing is we are using biomolecular biomaterials. So this is the material coming from the cell itself. We are using DNA, protein, carbohydrate, this kind of biomolecules to create some uh, drug delivery platform to deliver some anti-cancer drug or anti-diabetic drug in cells and proper locations. Also, we are interested in early detection of diseases, for example, cancer, whether we can detect this cancer in early stages before you have symptoms of this disease. And also, we are interested in how to convert carbon dioxide to some polymer out of this platform and reduce the carbon dioxide emission and use it for some product development. And also we are interested in fundamental studies of this assembly of these molecules inside the cells. And our research are currently funded by Department of Biotechnology and NSAR, um, and then IIT Bhilai also. So if you are interested about our research, what we are doing, you can visit our website. But briefly, I'll show you some of the data we are generating in our lab. Okay, so our lab is currently interested in artificial DNA nanostructure, right? So we have a natural DNA in our cells, so this DNA are inside our nucleus of our cell, but we are um, making artificial DNA nanostructure out of this synthetic DNA. So we have this DNA synthesizer in our lab, and you can make DNA of any length and any sequence from this DNA synthesizer. Okay, so this is chemically made artificial DNA and single-stranded DNA, right? So then this DNA can be grafted on any nanomaterial using suitable chemistry. So you have a nanoparticle, it can be plasmonic nanoparticle like gold or silver nanoparticle. It can be metal oxide nanoparticle like iron oxide nanoparticle. Can be quantum dot like zinc oxide, zinc sulfide, those kind of quantum dot. Or it can be polymeric nanoparticle, metal organic framework. Depending on your this nanoparticle, you generate different chemistry and you graft this DNA on the surface of this nanoparticle, right? So this is called DNA nanoparticle. Classically, it has been developed by Professor Chad Markin from Northwestern University. Or you can have another thing, you can have another long piece of DNA and you fold this short DNA with the long DNA, and you can generate different kind of DNA nanostructures. So these nanostructures are essentially made out of only DNA, okay, via the DNA duplex formation. I am not going into basics. I assume you know what is DNA duplex. If you don't know, you just go back to your textbook. So this is essentially forming a DNA duplex formation, and you can generate this kind of different DNA nano objects, and the diameter and length scale of these objects are less than 100 nanometer, right? So we can generate this kind of artificial DNA nanostructure, and you can use it for different purposes. For example, you can use them for understanding the chemistry and physics at the nanoscale, what is happening in your cell. Okay, so we call it nanobiochemistry and nanobiophysics. We can get plasmonic metamaterials out of this system. We can use it for electronics, nanoscale biosensing, drug gene delivery, super resolution imaging sequencing. All these things are coming up, and a lot of different labs all over the world, they are working on this. So in our lab, we are specifically interested in disease detection as well as the drug and gene delivery for cancer, right? So essentially what we are doing, we are making platform for cancer theranostics. So as you know that cancer is currently affecting a lot of people. I'm sure some of you are aware of your friends or family, they are suffering from cancer. It's a huge problem all over the world, right? So the thing is, when you have cancer, it is one of the disease which, if it is detected early stages, and if you have therapy, proper therapy of the disease, uh, then you can actually cure cancer, right? So that is not true for other different kind of aggressive diseases. For example, diabetes, you cannot really cure diabetes, or you cannot really cure heart disease. Okay, um, so early diagnosis and therapy, good therapy, is very important, so that the chances of survival gets improved, right? So these are current modalities of diagnosis of cancer. So when you have some symptom, doctor will tell you to undergo different kind of diagnosis, and based on the diagnosis, you can see whether you have cancer or not. So you have X-ray, ultrasound, scintillation, positron emission tomography, computed tomography, and so on. This kind of clinical modalities are available to detect cancer, right? And also, after this is detected, what happens is doctor will decide your therapy. What will be your therapy? Depending on your cancer, doctor will decide the therapy. So primary line of therapy is for solid cancer is surgery. So doctors will remove the tumor tissue from your body. Then you can undergo hormone therapy. Depending on your cancer, you have different options for a doctor, right? So you have hormone therapy, you have bone marrow transplantation for blood cancer, you have chemotherapy, so you can use chemicals or drugs to cure your cancer. You have targeted therapy, radiation therapy, immunotherapy. Those kind of things are available. Okay, so if it is detected in very early stage, that means your cancer is not spread throughout your body, it has not metastasized, then you can have surgery after detection, and then you can do all this, one of these therapy, so that your cancer don't grow. Okay, so nanomedicine, which is kind of emerging field, which combines actually the diagnostics and therapy. So this is a theranostics is a combination of therapy and diagnostics. So nanomedicine can do both this therapy as well as diagnostics in one in all platforms. So you need to have inject just one nanomedicine, then it can do therapy as well as diagnostics, right? So how do these nanoparticles go inside the tumor specifically? So when you have a tumor in your body, the blood vessel in the tumor actually are not well developed, right? So you have lot of pores inside the blood vessel. So in normal blood vessel, your nanoparticles cannot go through the tissue, and this nanoparticle size is more than 100 nanometers, so it cannot penetrate the blood vessel to the tissue. But when you have cancer tissue, these blood vessels are very leaky, and you can think these vessels as a leaky pipe. Okay, so these nanoparticles can go inside the tumor tissue. Okay, so this is called passive targeting or EPR effect. And also, if you have some molecules on the nanoparticle surface which will have very nice interaction or affinity with the cancer tissue, then also this nanoparticle can go through this blood vessel and go to the tumor tissue. So over the time, this nanoparticle gets accumulated inside the tumor tissue, not in the normal tissue, right? And if you have some molecule which can emit light or give the drug molecule embedded on the particle which can be released inside the tumor tissue, so specifically you can do this imaging and therapy together. Okay, so the advantage of this nanomedicine is, one, you can combine therapy and imaging in one nanoparticle, and second is it can specifically go to the tumor tissue, not in the normal tissue. So we have very high specificity of the tumor tissue. Okay, the second one is called active targeting, right? So this is just one example showing that this is the image showing that you have injected a nanoparticle that is emitting the purple light, and this nanoparticle goes to the tumor tissue specifically and lights up the tumor tissue in purple color. Okay, so this is blood vessels. These are again labeled by some fluorophore, but you can see that your normal tissue is not having any contrast. This is like a black, and this is purple. So you can actually differentiate between cancer tissue and normal tissue, right? So this is the one of the advantages of this nanomaterial. So actually, if you look at the anti-cancer therapeutics, there are several nanomedicine-based formulation that are coming out. So you can see, I have shown you some images of clinically approved nanomedicine. For example, this is doxorubicin HCl liposome. This is called Doxil. So this is a anti-cancer drug, and that is encapsulated in a liposome, which is in nanometer scale dimension, right? So this is typically 100 to 200 nanometer dimension. So then also you have this Abraxane, which is the albumin-bound paclitaxel, which is the anti-cancer drug. There are several formulations that came out in last 10 years, and a lot of research are happening in preclinical stages. So we are trying to develop something which will actually give idea to the surgeon or a oncologist where is the cancer tissue and where is the normal tissue. So this is a simple cartoon picture. So you have some tumor in the brain. So if you don't have any nanoparticle injected, this tumor tissue don't have any contrast with the normal tissue, and therefore during the surgery the surgeon cannot distinguish between normal tissue and cancer tissue. Okay, so it's very difficult to do proper surgery to remove all the tumor burden from your brain. So you need something which will actually differentiate between cancer tissue from the normal tissue. So this cancer tissue is glowing, that means we have injected some nanoparticles, and nanoparticles has gone to the tumor tissue specifically via previously mentioned mechanisms. And if you have a QR code, for example, attached to the nanoparticle, you can simply scan this QR code during the surgery. And QR code for cancer is different from QR code of normal tissue, and you can easily distinguish between cancer tissue and normal tissue. So this is a scanning technique. So you can scan everywhere in your brain, and you can look for the QR code using the QR code scanner, right? So this is actually very good strategy, but you can see that this brain is a very large area, and every point scanning of this QR code is not very efficient, right? So it will take long time to identify the QR code for the cancer tissue. So this is not very efficient. So it will be useful if we have something which will give you additional signal. So QR code with some additional signal will actually help the surgeon identify that, oh, I have a signal here, and let's see, and let's go there and identify the QR code. And if the QR code matches with the cancer QR code, this area will be resected by the surgeon, right? But the problem is this optical based technique suffers from deep tissue penetration is very low, right? So most of the time your cancer is detected in a very deep in your tissue, and this can be obscured by the tissue that is on top of your cancer cell, and that will obscure. So it will be nice to have some modality which will give you light from deep tissue. Okay, so overall we need to have three different modality. One is used for deep tissue imaging, second is used for very fast imaging, and third is this QR code. Okay, so for efficient theranostic agent, we need to combine these three different modalities. So that's what we did in this paper. So that appeared last year in Nature Communications, where we incorporated this QR code, which is the fingerprint-like spectrum from the molecule that is embedded on this nanoparticle. So this has a very unique spectrum, and this is actually the QR code for this molecule. Then we have a fluorescence, which is used for faster imaging and intraoperative guidance of the surgeon. And third is the photoacoustic modality, which is used for deep tissue imaging. So if you have some cancer which is deep inside your body, it can be detected by this photoacoustic modality. And additionally, we have incorporated a therapy modality. So there we are using photothermal therapy. That means you are shining light and generating heat out of these nanoparticles, and because of this high temperature, your cancer cells are dying. So this is typically called photothermal therapy. Okay, so we have a triple modal nanoparticle with these three imaging modality and one therapy modality. So this is a combination of imaging and therapy. So this is a theranostics nanoparticle. So let me just briefly uh, tell you about this two different modalities, Raman and fluorescence light, we are using in this nanoparticle. But if you understand a little bit of photophysics of dye, you can say that Raman and fluorescence light doesn't exist simultaneously. Okay, so in normal condition, fluorescence light is very strong and Raman light is very weak. Okay, so Raman light is very uh, high in intensity, at least 10 to the power 6 times higher compared to the fluorescence light is 10 to the power 6 times higher intensity compared to the Raman light in isolated dyes. Okay, if you have nanoparticle and the dye is far away from nanoparticle, this is the case. Okay, but when you bring this dye to the surface of gold nanoparticle, what you see is you have quenching of this fluorescence. So your fluorescence intensity goes down rapidly, and your Raman intensity goes up by a surface enhanced Raman scattering effect. Okay, so your Raman signal is now very high, and your fluorescence signal is very low in intensity. So to have a optimum Raman light as well as fluorescence light, we need to create some distance, which is a optimum distance window. So this is a typically 2 to 10 nanometer from the surface of the nanoparticle. That's where you need to keep this dye so that you can get a optimum fluorescence light as well as Raman light. Okay, so that is very tough to maintain this distance. So we are actually using a piece of DNA, small DNA length of around 5 to 10 nanometer, and depending on DNA sequence, you can change the distance between the fluorescence and the distance between the gold nanoparticle surface and the dye. Okay, so without going into much details about the design, what you see is different generations of this triple modal nanoparticle. So initially we created something spherical nanoparticle with a polythymine DNA sequence and didn't work so well. So the Raman intensity is very low and we could not detect this nanoparticle in mouse models, right? So we did some rational design optimization based on lot of theoretical inputs like molecular dynamics simulation, electromagnetic simulation, those kind of simulation we have done. If you are interested, I can talk offline how it is done and why it is done. But for the sake of the time, I just keep that portion. And I want to say, after the design optimization, what we found that if you have a gold nanorod core and you have a polyadenine sequence with this particular dye and a silver shell, you have much brighter fluorescence as well as brighter Raman out of these nanoparticles. Okay, so the limit of detection of around 5 femtomolar, which is very low concentration. Uh, so how do you check these nanoparticles whether they are actually giving you proper signal in reality? So we tested this nanoparticle in a mice model of cancer. So we have a ovarian cancer mouse model. So ovarian cancer, you can see in the tumor is generated in the mouse model, and we injected this mouse model, and you can see this is the photoacoustic data. After 24-hour injection, the spot was lit up by the signal. That means you have this cancer tissue which is visible from deep tissue environment. And then we did this NIR infrared fluorescence imaging. So left hand side you have the mouse which is not injected with the nanoparticle, and this is injected with this triple model nanoparticle, and you can see after 24 hours there is a this tumor tissue is lighting up. Okay, and you can see that contrast between tumor and background is getting very high. And then the surgeon can use this fluorescence light to detect this tumor tissue and actually guide the surgeon for the resection of the tissue. So you can see this surgeon has resected this tissue under a fluorescence camera, and you can see there is some residual fluorescence signal. That means you have some residual tumor tissue, and after second resection you can remove all the tumor burden from a mice. Okay, so without this fluorescence light you might have missed this residual tumor tissue in your body, and after the first surgery you go back home, and then this tumor tissue actually will grow over time and you will get a recurrence of the disease. Okay, so this is why you need to have complete resection of tumor from your body. Okay, so complete resection of tumor is done by this fluorescence signal, right? So what is the use of this Raman signal? So Raman signal is essentially used for verify whether this tumor tissue is actually true positive. This is the QR code of the nanoparticle. If it is present, is actually positive. So in fluorescence, you have a drawback of autofluorescence. So you have some low level of fluorescence from the normal tissue as well. You can see that the tumor tissue has high fluorescence and normal tissue has low fluorescence, but during the surgery, you don't know whether this low fluorescence is coming from the normal tissue or cancer tissue, right? Then you go to this individual tumor tissue and normal tissue, you scan the tissue, and you can see this signature of the nanoparticles that these are present in tumor tissue, these are not present in normal tissue. So essentially this is a QR code to verify whatever tissue you have resected, is it tumor or normal. Okay, and this is again verified by histology. I am not going into details, but you can see that if you have more density of cells, that means your tissue is cancerous. You have less density of cells, you have normal tissue, and from there you can actually verify uh, uh, your tumor is completely resected. Okay, and we have done all the different kind of fancy experiment where you can actually see this is the fluorescence image of excised tumor, and there is some high fluorescence from some area, there is low fluorescence from some area. Similarly, you have high Raman signal from some area, low Raman signal from some area. That actually corresponds to your H&E staining, which shows you the actual cancer tissue. So you have more blue means it's more cancerous, more pink means it's not cancerous. So it actually corresponds very well with the H&E imaging of this tissue. That means this is actually very nicely correlating with the gold standard, which is H&E. And as I told you before, this these leaky vasculatures are actually responsible for leaking this nanoparticle inside the tumor tissue. And if you do high resolution fluorescence microscopy, you can see these green spots are actually in the vessels, blood vessels, are red spots are actually this nanoparticle. And you can see these nanoparticles are coming out from the blood vessels inside the cancer tissue. Okay. Okay, so let me show you just one more example of this kind of imaging. This is imaging in different cancer model. This is called glioblastoma multiforme, or GBM. So this is actually one of the most aggressive brain cancer that is available, and you need to undergo surgery immediately after you detect this brain cancer, right? So you can see that this mouse has grown significant brain cancer in the MRI image. The white spot means there is some cancer in here. And then we injected the mouse using your triple model nanoparticle, and you can right away see after the injection there is some signal coming from the brain. So that means there is some problem in the brain. Okay, and you excise the brain tissue and you can see the hot spot, and that actually corresponds very nicely with the cancer tissue that is detected by H&E imaging. Okay, so the fluorescence hotspot and Raman hotspot is actually corresponding very well with the H&E imaging. So we are very confident that our probe actually goes to the location of cancer and lights up the cancer, and we can detect this cancer in three different ways. One is using uh, photoacoustic modality, second is using fluorescence modality, and third is using Raman modality. So we are very confident, we are very accurately can detect this cancer tissue in any animal model, and we are looking for translation of this kind of nanoprobes. Okay, let me go to very briefly about the photothermal therapy. So as I told you before, these nanoparticles are made out of gold. So if you shine a laser light, this photon energy will be converted to heat energy by these nanoparticles. Okay, so what we see is here we have in the bottom row we have this mice which is injected by phosphate buffer saline, which is just the regular salt water. Okay, and top mice is injected by these nanoparticles, and as tumor, and we shine a NIR laser on the tumor, and what you see is over time your tumor heats up. So this is actually the thermal imaging. Okay, so this is same as the thermal imaging you can undergo in different airports or in different malls. So this camera measures the temperature of cancer species. Okay, so over the time we are measuring the temperature of the cancer area, and you can see the temperature of the cancer area increased over 2 minutes. So it increased to 55 degrees Celsius. In PBS injected mice, this temperature didn't increase as much as the uh, nanoparticle injected mice. So here is a huge increase in temperature in this area. Okay, so that leads to destruction of the cell surrounding this area, and mostly these are cancer cells. Okay, so as you can see that these mice all are having cancer, but these mice injected by nanoparticles uh, got the photothermal therapy, and the other mice were controlled. So this mice was injected by nanoparticle, didn't get photothermal therapy. This mice got injected by salt water, got photothermal therapy, and this is only control mice with only salt water injection. And we did photothermal therapy in these two mice and didn't do photothermal therapy in these two mice. So you can see nanoparticle with photothermal therapy, this tumor didn't grow, and eventually this tumor didn't grow and this mice got cured. You can visually see, and the volume of the tumor got decreased, you can see. But all the other groups, you can see that tumor volume is increased, and over time it will metastasize in your whole body and the mouse will die. Okay, so you can, using this nanoparticle with photothermal therapy, you can actually cure cancer. Okay, and what you see is this is the standard histology test we do. So this tumor is showing the cancer tissue, but if you take tissue from this area, you can see all this cancer tissue is actually dead. Okay, so more pink means you have like necrotic tissue or dead tissue, right? So our triple motor nanoparticles show excellent photothermal therapy outcome. So we can use it for imaging as well as therapy. And last year it was published in Nature Communication. So then we started to understand this gold nanoparticles might not be very beneficial for us because number one, gold is expensive. Second is gold might not be biodegradable, right? So after you inject this nanoparticle inside your body, this gold can go to your liver or spleen, this kind of organs, and although it's not toxic to your body, but long term it can create some problem, right? So we wanted to have something different from this gold nanoparticle based platform. Therefore, we wanted to have something which is biodegradable. Uh, so the model we chose for this kind of study is this triple negative breast cancer because triple negative breast cancer is very hard to cure. You have heard of that. Is very one of the very like aggressive cancer, and the therapy of this kind of cancer is limited, right? So recently, literature says that overexpressed in this kind of cancer. So we can see that folate receptor are nicely overexpressed in negative breast cancer compared to other breast cancers like HER2 positive breast cancer or ER positive breast cancer or normal breast, right? So you can selectively target this triple negative breast cancer using folate receptor targeting. So for that, what we have is this DNA origami, which is made out of this staple strand and this a long single-stranded DNA. So this can be folded together to form this DNA origami, and you can place this folate on some particular DNA strand, and you can place this imaging moiety, some near infrared dye on this nanostructure, and under the AFM image you can nicely see this triangular shaped DNA origami is formed, and size of this is around 50 nanometer. So the scale bar is 50 nanometer, so the one arm is around 120 nanometer. So it is formed nicely, and then we checked this targeting of this triangular origami in two different cell line. One is MDA231, one is 468. You don't have to remember these names, but one is low folate receptor 1 expressing, one is high folate receptor 1 expressing. 468 express this folate receptor protein very highly. So you can see after 4 hours and 16 hours, our DNA origami probes entered this folate receptor positive cells better than the folate receptor low expressing cell. So you can actually target this high expressing cell using our platform. So then we wanted to test in vivo. So after the in vitro study, we injected these cells and grow the tumor. So left hand side you have folate receptor positive tumor, and right hand side you have folate receptor negative tumor. Then we injected our this DNA origami, which is folate receptor targeting DNA origami, and you can see from the imaging, this left tumor lights up more than the right tumor. So this is the ex vivo means you have resected the tumor out from the body of the mice, and you can see this left hand side tumor is having higher fluorescence compared to the right hand side tumor. Okay, so that means you are actually targeting the folate receptor positive tumor. That is also shown here. I am not going into details, but you understand that left tumor is expressing the folate receptor 1. More brown means you have more folate receptor 1. Okay, so left hand side, since this is folate receptor positive, it got more signal compared to the right hand side, which is folate receptor negative. Okay, and you can easily target this kind of cancer using our platform. Okay, um, so let's next. How much time do I have? maybe 10 minutes. Okay. So let me just quickly show you some of the recent data. What we are doing is we are using hydrogel for drug delivery. So we all know hydrogel. What is the hydrogel is? Hydrogel is a polymeric network, and in this network we have a very high water content, up to 80 to 90% water. Okay. So therefore, this polymer can be modified with some drug, and then you can implant this polymer inside a body, and this drug can be released over time. And if you look at the different length scale of this polymeric network, it will look like a gel, but if you have higher magnification microscope, you can see that there is a polymeric mesh, and in the polymeric network you have this water molecule that are residing, and you have this drug. Okay. So we wanted to use this platform for delivering drugs for diabetes. As you know that this is we are talking about a pandemic COVID-19, but in long term diabetes will be a worldwide pandemic, right? So right now we have 422 million people that are infected, that are having they are having diabetes. Okay, they don't have any other choice, and over the time. um because of the diabetes, you get different problems in your eyes, in your heart, in your kidney, and so on. And this is a huge problem in the third world country as well as in the Western world, right? So let me show you why what we are doing and why we want to do that. So right now, what is happening is what is this diabetes? What is happening in diabetes? In normal person, what you have is after meal, you will have digestion of the food, and you will increase your blood glucose level. And after some time, this blood glucose level will come down because of this production of insulin. Okay, so you produce the blood glucose, and then this will trigger the production of insulin, and insulin will increase the glucose metabolism rate in your cells, and the blood sugar level will come down very easily, and it will be having a very low blood sugar level. Okay, the average blood sugar level is around 120 cc per mg, right? So what happens is in diabetes patient, this feedback between the glucose increase in the blood and the production of insulin is disrupted. Okay, and you don't have enough production of insulin. So right now, what is happening is if you have blood glucose sugar level and there is no production of enough insulin, so your blood sugar level don't come down to um, your base level, and there is an increase of average blood glucose level. Okay, so right now what people do is they take drug, but most of the time drug will fail after a certain time, and you have to take this natural molecule insulin um as a drug, right? So people do the injection of insulin in your body. So before the meal, they take injection, and hopefully you'll have enough food, and then this blood sugar will go down because of the insulin injection. And most horrible thing about this insulin therapy is you have this kind of uh, pinching every time you take insulin shot, and then sometimes your blood glucose sugar level goes down below the average blood glucose sugar level, which is called hypoglycemia, and that is actually very, very fatal. Okay, and people might die because of the hypoglycemia immediately after maybe half an hour. People will die because of the hypoglycemia. So we need to create a platform that will actually mimic the natural production of insulin, right? So a lot of people are actually working on making artificial insulin or smart insulin. So this is typically made of polymeric formulation where you embed your insulin inside the polymeric network, and this insulin is not active in your body. But when you have production of glucose. You have a lot of glucose in your blood. This material will actually dissolve by the glucose, and the insulin will be released, and therefore this insulin can reduce the blood sugar level. Right. So this is called smart insulin. So this a lot of people are actually working in this domain, and currently the drawback is whether we can make a biodegradable or biofriendly material, this scaffold material for insulin. Right, most of the time you use natural, like artificial polymer, and that might be toxic to your body. So right now what we are doing is we are trying to use a natural polymer, which is the DNA, and this DNA is made out of this kind of shape. So this is a DNA origami, and then we are trying to cross-link this DNA origami and encapsulate this insulin inside the DNA origami three-dimensional network. So this is your hydrogel network, and then when it sees the glucose, this will react. This will open up, and it will react with this glucose, and then this insulin will be released. So the chemistry I'm not going to go into, but you can see that this kind of linker molecule that actually cross-link this individual DNA origami into a hydrogel, and then after your glucose is introduced in the system, this 3D network is disrupted, and then you can see the insulin is coming out from the network. Okay, so I'm just going into just one data where we have developed this kind of system. So this is in a lab scale, in a test tube. What we are doing is we are making this hydrogel uh, made out of DNA, and then we are testing this hydrogel, exposing this hydrogel into different concentration of glucose. So typically your blood glucose is around 7 millimolar blood glucose, and you can see over 60 hours of time, if you have blood glucose level more than 10 millimolar, uh, all the gels are dissolved. Okay, and the protein inside, insulin inside these gels are released upon the exposure of this glucose. Right. So we are getting very nice results in test tubes in the laboratory. So right now we are trying to collaborate with biologists and clinicians to use this nanoparticle in mice models and later in humans, so that will have glucose responsive insulin release. Where you need insulin, then only the insulin is produced. You don't have to inject insulin every time you take food, right? So this is the goal, long-term goal of this project, and we are making significant progress in this direction. Okay, so I think I'll stop here. So I think I have conveyed you what importance of the biomaterials research. So if you want to improve your lifestyle, if you want to improve your lifespan, you have to invest on biomaterials research. And as a researcher, I can say in India, very little research is going on compared to Western world. So we need to invest more money on biomaterials research so that we can live happily and live longer, right? And also, I have shown you some data on DNA nanotechnology based cancer diagnostics agent that are nicely used for surgery and therapy of different kind of cancer. And lastly, I have shown you some data on DNA hydrogel, which is developed for smart insulin release. Okay, and we are looking forward to more funding and more collaboration for clinical translation, and hopefully over 2-3 years in new campus we can have translation of this product, which is, I've told you before that it takes time, but we have to be active to do this clinical translation of this product. So with this I'll stop, and this is the picture of the campus we are going to have very soon in Bhilai. And I want to acknowledge my funding agencies as well as my students in the lab. They work very hard in these situations. In COVID situations, they are continuously working and generating data. I'm thankful for them, and I'm thankful to IIT Bhilai for giving me this opportunity. And also thank you for listening and I'll be happy to answer any question you have. I'm sure you have a lot of question because this is not very familiar to you, but I'll be happy to answer any question. Right. So I stop here and look forward to question.
Okay, thank you so much, sir, for sharing your valuable knowledge with us and with our participants. Uh,
Okay.
so there is one question in the chat box.
Okay.
I request participants to please ask their queries.
Okay. If they are not they are shy to ask question, they can write in the chat box. I can read.
Yes, sir. Yes, sir, you can, participants, you can ask the query in the chat box also,
Yeah, I
while
can read the
using the
question.
mic also.
Yes.
So there is one question by Vishakha Singh. Uh,
I didn't understand the question properly.
yes, it's confusing. EDU in vivo mouse troubleshooting.
I think is
Means,
means okay. Maybe Vishaka can explain the question a little bit more. Uh, but yeah, we do little bit of collaboration with lot of biologists to understand our materials. And if you want to do clinical translation of any biomaterials, actually you go for animal testing, in vivo testing. That's what I told you. Uh, Only working with cells are not sufficient. So you need to test your material in vivo, right, in mouse or in larger animals. So we are currently building the mouse facility, and maybe in 2 years we'll have full-fledged mouse facility in IIT Bhilai campus. Uh, but you understand, when you want to have the biomaterial in your body, you don't want to have a untested biomaterial in your body. Of course not, right? So you need to have some model. Okay, although you are giving lot of pain to the animal, but it's worth it, right? Before testing the material in your body, you need to test the material in body of some mice or some small animals. Okay. There is another question that comment on potential uses of nanomaterials for targeted drug delivery. Okay. Okay. So this is the current active area of research. So I'm also okay. I am also doing some research on this. So a lot of people are doing this kind of research because this has huge potential. Okay. So huge potential means it has potential showing that it can go into cancer tissue very efficiently and we can image it. But it has also has drawback. I have shown you good pictures, but the drawback is, so number one drawback is the nanomaterials also goes to other organs like your spleen, liver. This kind of organ likes to take these nanomaterials, and if your nanomaterials is not biologically like not very biofriendly, this organ might get damaged, right? So if you want to cure cancer, you might not want to create damage in your liver or spleen or kidney, something like that. Okay, so there is a drawback as well as advantage. So people are working to solve the drawbacks of this nanomedicine, and there are active research going on to how to bypass this problem, right? So it will take some time, and you can see that a lot of nanomedicine that are coming to in the clinics and in the real life. You are currently using nanomedicine without knowing that it's a nanomedicine. Okay. Okay, Bhavesh Sinha, he's asked that can DNA origami you are targeting be able to hold any other drug as well? Okay, Bhavesh, this is a nice question. Yeah, we incorporated drugs, and I didn't have time to show you all the data, but the paper is already submitted. So we actually incorporated a drug called doxorubicin in the DNA origami structure by some chemistry, and then this is delivered to your cancer cell, and we saw there is a significant delay in cancer growth in case of DNA origami with the drug compared to without the drug. Okay, so Vishaka Singh asks, EDU means, okay, so this is a typical anti-cancer drug, EDU, right? So yeah, I didn't work on this kind of drug. This is very specific drug, but in general, if you can attach any drug to your nanoparticle, since we know these nanoparticles goes to your tumor cells, goes in higher amount in tumor cells compared to the normal cells, normal tissue. What happens is your drug is also going to target towards cancer tissue compared to the normal tissue. The drawback of this chemotherapy is what when you have a chemotherapy, you can see a lot of side effects. Yeah, so a lot of side effects are there because of this side effects comes from. Yeah, can everybody mute? I can. Okay, so this nanomedicine specifically goes to the tumor tissue. Only small molecular drug that will go to your normal tissue as well as tumor tissue, and that will create side effect because currently you see the chemotherapy. The problem is you have side effects like you have side effects like you lose your hair, you lose your taste, something like that. And most of the death due to the cancer is due to the side effect of the chemotherapy. Okay, it's not due to the actual cancer, right? So we have to reduce the side effects so that uh, the therapy is more viable and less toxic to your body, right? So you can, in general, attach any drug molecule to your DNA origami structure. In principle, you have to develop a chemistry to attach drug. Then you can deliver it specifically to your cancer tissue, right? Okay. So which type of test is needed for any material for biomedical application? Okay, so there are different kind of tests. So depending on what is your biomedical device is going to do. Okay, so let me give you one example. If you have, if you want to do a drug delivery platform, right? So you want to test whether this material is not toxic to your body. This material is not creating any problem to your blood because it's going to be circulating in your blood for a long time. If the material is creating like thrombosis or creating clot in the blood, that is not also good. So uh, cytotoxicity study, and then there should be no immunogenicity. So that means your immune system should not be responding to your medicine. And then a lot of different kind of study, genotoxicity, those kind of studies are there. So depending on how you are going to use this, so biomaterials is a broad class of material, right? So different classes of material have different testing. I cannot pinpoint just one test or two tests. It's a spectrum of test. Okay. Okay. Kanchan Devangan, he is asking which type of test. Okay, I answered this question. Vishal is asking how far these structures are from artificial virus. Okay, that's an excellent question. So artificial virus, they are three-dimensional spherical structure and different kind of shapes that can be there. For example, if you have a coronavirus, this is a spherical virus with lot of spike proteins. Okay, so right now these DNA structures are can be made into different shapes. Uh, there is a design platform and design software. You can design different shapes and you can make DNA origami structure. And fine details you need to verify, and experimental in that needs to be verified. Right. So that is current challenge. How far we can go to natural virus? It's an open problem, and a lot of people are working on this direction. Right. Okay. So the problem for biomedical researcher is it's funding. So it's very like expensive research. So you need to have good source of funding. You are not doing the research in your computer, you are doing research in lab as well as with animals. So animal experiments takes a lot of money, expertise, and so on. So current problem is lack of funding, but it's coming. We are fighting. We are writing grants and we are asking for money. So we can do whatever we can do. We can do in the lab scale, and then we can collaborate with some big pharmaceutical companies, and then we can bring our product into market. So that is the question. Good question. Like as a researcher, it's not only you publish good paper, that is not enough, because you are spending your like taxpayer money on your research that is uh, might not be useful for anything, right? So that is also required, but you need to develop something which is useful for some problem solving, right? So our lab is specifically interested in translation of our product, not only developing a product and bringing the product to the general public. So it takes lot of time. So in vitro testing takes few years, and in vivo testing takes another few years, and then clinical translation. And if you fail in between, you have to restart from the beginning. So it takes a lot of time, maybe whole lifetime. But if you bring one product in the market in your whole lifetime, that is your achievement. Okay. Okay, thank you very much. I think if you have any question, you can just directly email me. We can have a chat by phone. There is no problem about that. We can have another discussion maybe in future conferences. Right? Okay,
Okay,
thank
thank
you.
you. Thank you, Dr. Pal.
Okay, thank you very much. Thank you
Yes.
for the invitation.
Okay, thank you, sir.